Thank you. So yeah, I'm Struan. I'm currently doing Masters by Research at the University of Edinburgh, and I'm focusing on the use of digital endocasts to investigate the evolution of the Dipnomorpha. So the Devonian saw the diversification of the major groups of sauropterygian fish, giving rise to several different independent evolutionary trajectories, and an opportunity to examine the evolution of one of these trajectories and the sensory system in that trajectory is found in the Dipnomorpha. An examination of this group is quite useful as they are the direct sister group to all tetrapodomorphs, and we can use their extensive fossil record to look at the evolution of the brain and draw comparisons between these trajectories. So my current research focuses on the Porolepiforms, which are the sister clade to the lungfish. The Porolepiforms are poorly understood relative to the more well-known Dipnoe, and as such present an opportunity for generating new phylogenetic information and also determining the polarity of dipnomorph characters. Pauichthys currently sits on the stem leading to the Porolepiforms, as it shares a number of features with both the lungfish and Porolepiforms, but its status there is actually debated a little bit at the moment. Porolepiforms are then divided into two groups, the Holotychiidae and the Porolepidae. The Holotychiidae are accepted as a clade of more derived Porolepiforms, though the interrelationships are quite poorly understood. However, the status of Porolepidae as a clade is actually up for debate because they may just be successive branches on the stem leading to the Holotychiidae. So recently, specimens of Glyptolepis, one of the Holotychiidae, have been accessioned to the National Museum Scotland. This is the dorsal view here, and ventral view, and the snout is at this end in these images. And this is the posterior view with the top, the bottom, left and right, and the brain case would have sat in this space here. These bones, the parietals in the skull roof, and also the ones that sit behind them, the postparietals, are of particular interest because when they're preserved in 3D, they can show the brain and the sensory structures. So along with specimen already present in the NHM, this presented an opportunity for CT scanning. So by studying the 3D images of the internal space of these fossil crania, called the endocasts, we're not only given an indication of sensory abilities, but also create a wealth of morphological data that can be used to study these early vertebrates. For example, Clement and Alberg, Challens and Clement et al. Have all published detailed accounts of various Devonian lungfish endocasts. Here's the first one from 2014, and another 2015, 2016, and then here's a fourth one that's not been published yet. And these you can see quite clearly how different all of these structures are. And this is just within the lungfish, so as yet there hasn't been a digital endocast of a Porolepiform. So the aims of my project were to describe the endocast of a Porolepiform Glyptolepis and compare it with the anatomy of other Porolepiform reconstructions and lungfish endocasts. To obtain these, two fossils of Glyptolepis, one from the NMS in Edinburgh and one from the NHM, were micro-CT scanned. And this generates topographical images showing slices through the fossils. And in the program Materialized Mimics, you can segment these and then generate a 3D image to give you the endocast. And these endocasts can allow lots of further problems to be addressed, but ones I was looking at were the definition of dipnomorph endocranial characters and the polarity of those characters, but also investigating homoplasian endocranial characters within the sauropterygians. So here you can see the endocast of Glyptolepis possidens in a lateral view, alongside a reconstruction of a different Glyptolepis species from 1972. The digital model generally agrees with Jarvik's interpretation, but there are far more descriptive characters that you can now see in the digital endocast. I'll draw your attention to a few in particular. Hidden in behind these canals here, there's a ventral expansion of the endocast in a region of the brain called the Telencephalon. Also in the forebrain, the olfactory bulbs are sessile, meaning they sit in the front of the brain, as opposed to on stalks between the main body of the brain and the nasal capsule, which is up here. And this lends evidence to, there's a recent suggestion that the polarity of this character needs to be entirely reversed, with this sessile state actually being the primitive state. Also very noticeably, there's this really large anteriorly directed hypophysio recess, which you can see also in Jarvik as well, but you can see it in a lot more detail now. So I'll go through these in turn. But here's the other specimen in dorsal view this time. There was a fair bit of damage in the main body of the brain and down the right olfactory tract. However, the preservation in the rostrum and in the skull roof was pretty exceptional. Particularly, the profundus branch of nerve 5 is observed to have two main anteriorly directed branches, one of which bypasses the nasal capsule, it's this one here, and the other larger branch actually enters the nasal capsule before coming out of it again to rejoin the dorsal branch. But this dorsal branch exhibits a very similar morphology to the lungfish Dipterus, although Dipterus lacks the medial branch that enters the nasal capsule. So I'll discuss this configuration in a moment, but first I'll overlay the network of canals in the skull roof. So as you can see, this is a very complex network of canals, but importantly we can see a structure called the lateral line, which was involved in mechanosensory function running along here, to which a lot of these canals are linked. But these complex canals are important as they allow identification of that profundus branch that runs forward, because it joins up in this little complex here. The lateral line system of Glyptolepis, also interestingly, has some branches that connect to canals that are obviously not part of the lateral line system. So this complexity raises a whole bunch of interesting ecological and evolutionary questions, but that's actually out with what I'm currently doing. So the ventral expansion of the Telencephalon of Glyptolepis is an interesting find, because a trend of increasing expansion is known within the lungfish, with the notion that the lack of expansion is the primitive state and the derived is expansion of this region. Dipnomorpha, which is an early Devonian lungfish, displays a small degree of expansion, as does this endocast of Glyptolepis. So this could have resulted in a couple of ways. Either this expansion is autapomorphic for Dipnoe and for Glyptolepis, or it actually has its root at the beginning of the Dipnomorpha and then was subsequently lost in Poichthys and Jungolepis. This region of the brain is called the subpallium, and it's associated with sight and smell. So expansion in this region corresponding with an increased sensory input may make this ventral expansion of the telencephalon a fairly common trait to evolve, which would result in homoplasia all over the phylogeny, which could also be true of differences in the lateral line, with different sensory requirements corresponding to different ecological niches, altering the morphology of the mechanosensory organ. But observation of this trait in Glyptolepis, the telencephalon, highlights the need for care when assigning these states these characters as primitive or derived. One of the notable features of the endocast was the large anteriorly directed hypophysiorecess, and this is a trait that's shared only with Poichthys, as you can see there, and it's indicated by a green star on this little phylogeny. And these genera are to a lesser extent similar to the Onychodont Chingmonodis, Dipnomorpha, which is that early Devonian lungfish, but also to the tetrapodomorphs, Gorgonasis and Euthanopteron, and also the basal bony fish, Zarolepis. So these are denoted by the blue triangle on this phylogeny, and these possess a ventral expansion of the hypophysiorecess, going simply straight down. The third state is observed in Jungolepis and all Dipnoe other than Dipnomorpha, and they actually have a really shallow and small hypophysiorecess that doesn't extend down very far. So this distribution is suggestive that a ventrally directed hypophysiorecess is primitive for Sarcopterygians, with Porolepiforms and lungfish displaying two alternative derivations. But Dipnomorpha poses a bit of a problem there, because it would appear to retain the primitive state, despite Jungolepis being a more basal taxon, exhibiting a derived state. So again, there are issues surrounding the polarity. So this is a diagrammatic representation of the nasal capsule of selected sarcopterygians. At the top is the tetrapodomorph Eusthenopteron, in which the profundus is tiny and enters the postnasal wall just up here. Next is Jungolepis, which displays the circumvention of the nasal capsule as it passes over medially. And that's the same configuration as in Dipterus that I showed in the Endocast comparison earlier. Poichthys and Porolepis both have very, very large profundus canals entering the nasal capsule back there, while Glyptolepis appears to possess a mixture of both of these configurations, with a huge canal entering here, but also this medially bypassing branch going along here. So this seems to make sense with regard to the most closely related Poichthys and Porolepis, but the similarities between Glyptolepis and Jungolepis and all the lungfish is quite an interesting but also puzzling find. So here, ticks indicate that the profundus circumvents the nasal capsule, while the cross indicates that the profundus enters the nasal capsule. So we know that in primitive sarcopterygians like Chigmonodus have circumvention of the nasal capsule, but we also observe this in Jungolepis and Dipnoi other than Dipnorhynchus. In Dipnorhynchus, as well as the tetrapodomorphs and Poichthys and Porolepis, enters the nasal capsule. Then Glyptolepis possesses both of these. So this distribution again could be a result of a whole number of different situations, but the most parsimonious explanation is that the common ancestor of all dipnomorphs here, but perhaps also all tetrapodomorphs as well, developed an autapomorphy which was subsequently reversed in Jungolepis and the Dipnoi other than Dipnorhynchus, but then Glyptolepis developed a sort of secondarily derived trait. But the superficial similarities between Jungolepis, Dipnoi, also Glyptolepis, and primitive sarcopterygians raises some questions about homoplasia and the definition of polarities based on this trait. So this situation in the nasal capsule and the ventral expansion of the Telencephalon, they reveal that polarity and definition of these endocranial characters is much more complex than we've thought up till now. Importantly, these questions have been raised through investigation of just one taxon, and with so many endocasts now emerging, there is potential for false interpretations based on these endocranial characters. So therefore, if possible, endocast characters and databases should be improved, polarities confirmed before we conclude meaningful phylogenetic results from them. And this has wider reaching implications for the definition of endocranial characters, as some may need to be revised, some may prove to be uninformative, but others may just be informative within certain groups like the Dipnoi Porolepiforms, but they actually fall apart at larger scales. The endocasts of some basal dipnomorphs and more crown Porolepiforms should help to clarify the polarity of these characters. But along with this, the new virtual study of previously studied taxa like Jungolepis may reveal hidden features and lead to revision of characters as well. But until we have a better understanding of endocasts, we should take great care when assigning character states as primitive or derived, but also when including endocranial characters in phylogenetic analyses. So I'd like to thank SVPCA for giving me the opportunity to present this work, and also for giving me the Jones-Fenley Memorial Fund, which enabled me to be here. I'd also like to thank the co-authors, particularly Tom Challens, for his help with the presentation and work. Thank you.